We will start in 25 seconds.
I'm Rachel Armstrong, Professor of Experimental Architecture at Newcastle University and Coordinator for the FET Open Living Architecture project, which ran from April 2016 to September 2019. We're a 6-partner-strong consortium, including Newcastle University, the University of the West of England, University of Trento, Spanish National Research Council, Liquefer Systems Group, and Explorer Biotech. Living architecture is a modular bioreactor wall that processes waste. It aims to change building impacts, which are a worldwide challenge and account for around 40% of our carbon footprint, which is a bit more than transport. Living architecture achieves this by altering the effect of our domestic lifestyles on the environment, where constituent households do not just consume resources, but also generate housework, transforming the notion of waste. Right now, the best we can do in terms of energy consumption is net zero impact. If we include on-site renewable energy sources, we might even put some energy by or trade with it on the power grid. Energy use, though, is only one aspect of our lifestyles, and net zero solutions do not address the broader issue of resource consumption. Whatever way we produce energy, if we continue to consume at our current rate, then we'll keep on impacting the environment negatively, as nothing will temper our consumptive lifestyle patterns. By applying 21st century biotechnical insights to regulate the robust processing power of microbes, living architecture enables us to give something back from our waste, which is turned into usable resource again. Specifically, it produces energy in the form of bioelectricity, as well as cleaning water, removing waste, neutralizing pollution, reclaiming phosphate from washing liquids, and producing particular forms of biomass, all of which can be circulated within the home, as well as being good for the environment. Living architecture is so much more than a multi-utility system. It is the foundation for a circular economy of the household. The key question that living architecture therefore raises is just how far can we design and direct the microbial processes within waste to perform housework? Our consortium set out to prove this principle by bringing together the insights of metabolic networks, synthetic biology, genetics, bioenergy, bioelectronics, informatics, and design to produce a freestanding interior partition wall about the size of a large bookcase, suitable for installation in a bathroom or kitchen. The wall is made up of bricks. These double up as integrated building blocks, where each brick is also a next-generation selectively programmable bioreactor and ideal home for microbes. 3 different kinds of bricks are used in the wall: the microbial fuel cell, an algae photobioreactor, and a synthetic consortium bioprocessor. Each type supports different kinds of microbes, and together they are assembled as modularly stacking, standardized building segments. Liquid domestic waste in the form of urine and grey water, is used as a feedstock, which is sequentially passed through living architecture's various chambers. As a living structure, the wall's operations can be likened to the workings of a cow's stomach. Each brick differentially processes liquid and organic matter within its gut, changing the substrates they receive into a range of metabolites that are moved on to the next chamber where further transformations take place, and so on. Each brick can be assigned a particular task, which is possible by altering the microbial populations and then sequencing them spatially. In other words, the system is programmable and can be thought of as a metabolic app. Two basic types of metabolism are used in living architecture. One is anaerobic, based on a technologically mediated composting process that can be harvested for electricity, water, and oxygen. As a standalone unit, it's known as a microbial fuel cell, which is a kind of battery that's powered by organic waste. The microbial fuel cell consists of an anode, selective membrane, and cathode. The hardware captures electrons from the bioelectrical activity of the biofilm that forms on the membrane to provide small amounts of usable electricity. At the same time, the fuel cell cleans water and produces other metabolites. The system is self-powered, and its actions are coordinated by an AI, which detects the amount of electricity produced by each brick and regulates the inputs accordingly to optimize the system outputs. The other metabolism is photosynthesis, which is carried out by tiny green organisms, or algae, in a photobioreactor, which traps sunlight and carbon dioxide that is used to produce solid biomass. Our system is gravity-fed, with a unique geometry and triple baffle system that promotes mixing. The main challenge of the project is to bring the different systems together so their various products can be meaningfully exchanged. We achieved this by boosting the electrical power of the microbial fuel cells by introducing oxygen from the photobioreactors into the cathode as a powerful electron acceptor. Achieving this integratability changes the very notion of waste, because the outputs of one system can be used directly by another, generating a cyclical economy of matter powered by metabolism. The final bioprocessor type that we developed is a synthetic bioprocessor, which examined the biological flexibility of the metabolic apps, helping us establish just how far we can design the actual metabolic reactions using synthetic biology techniques. While our modified system cleaned nitrous gases from the air, our indicator product was organic phosphate that was reclaimed from detergents. Inspired by the configuration of the microbial fuel cell, the synthetic bioprocessor was made up of a farm and labour module, separated by a selectively permeable membrane. The farm system feeds the labour module with a rich supply of carbon from organisms that overproduce sugar. This passes through the membrane into the labour module and reaches workhorse organisms whose genes are well known. The sugar-rich environment enables the design of new metabolisms as under these conditions, enabling us to mix and match genetic sequences as a mixed population of modified microbes forms in ways that are not possible in nature. Specifically, these new metabolisms can reclaim phosphate from detergents and remove polluting nitrous gases from the air. The ability to stitch different metabolic building blocks together between microbial species greatly increases the metabolic range for design and enables us to work with these processes at scale. During the project, we created 2 new genes, one with a patent granted and one pending. We also developed a new software called Dulix, which is a web-based DNA library design tool that makes gene sequencing more intuitive. The new knowledge produced in this project also secured further funding in new grant applications for environmental remediation and architecture, multiplying the revenue generated to around 10 times that of the original €3.2 million investment. Throughout the project, we also considered the social acceptability of the technology through various brick prototypes that were exhibited in biennales and international exhibitions. The first prototype was a simple hack of a brick, turning it into a microbial fuel cell, which brought together structure and process. This was displayed at the Building Centre in London in 2017 and during the Venice Architecture Biennale in 2017. We also made structures that could simultaneously host unlike environments, such as this brick designed for the 4th Tallinn Architecture Biennale in 2017 that links together photosynthetic and anaerobic organisms, enabling them to exchange metabolites with each other. Our final brick prototype, based on this principle, contained 4 chambers separated by ceramic plates, where we reclaimed 100% of the phosphate that was introduced into the system. And this diagram shows how the spatial sequencing of the Living Architecture project was configured, thereby completing the metabolic app. The final wall shown here used synthetic organisms, so it was explored in a laboratory rather than a social context. Although we couldn't use the original Living Architecture wall in a public setting, we generated a prototype system that was shown at the Whitechapel Gallery in collaboration with artist Cecile B. Evans as part of the Is This Tomorrow exhibition. The installation is an apartment space where a screen system is powered by wild-type organisms within a set of microbial fuel cell bricks. The work can be thought of as a post-human household, inhabited by ghosts of the past, present, and future. The uptake of living architecture raises important questions about how we live. For starters, we must question the need for a 230-volt electrical main supply within our homes. Given that we step down a lot of our electrical power, the prospect of 12-volt lifestyles is raised, an amount that could be reasonably provided by the living architecture infrastructure. Stepping down our power supplies does not mean a less advanced lifestyle, but one that is experienced differently. In the video, we demonstrate that microbial fuel cells in the living architecture system can power a window-opening robot. Rethinking how we perform housework opens up new frontiers in low-power electronics and robotics and invites an era of innovation in low-power domestic appliances, such as using ultrasound to clean our clothes or employing thermocaloric materials in refrigeration. Tim Garrett, an atmospheric scientist, Matteo Scriselli, a mathematician, and Stephen Keen, an economist, have recently looked at the economy through a thermodynamic lens. They concluded that although current efforts to reduce greenhouse gas emissions are focused on improved energy efficiency, this has the side effect of making it easier for civilization to grow and consume more. They suggested the way forwards is keeping the economy at a steady state, where the energy we use is devoted to maintaining our civilization and not expanding it. Additionally, previous fossil fuel-based innovations must be phased out while innovating new non-fossil fuel technologies. A platform for a circular domestic economy that enables us to reconfigure our homes and cities, living architecture can help achieve the European Green Deal with a new thermoeconomics as the foundations for a regenerative society. Forming the basis of an economy that works for people, its operations can be customized and implemented in ways that promote the European way of life, which includes fairness for the natural world. To build an uptake community, we are developing a digitally enabled version of a brick to increase the usability of living architecture. This active living infrastructure controlled environment, or ALICE prototype, is supported by an EU Innovation Fund and provides a relatable digital experience that is powered by waste and communicates how microbes can perform valuable actions within our homes using real-time energy data to produce animations and online feedback loops that reinforce how we can help them do this. Supporting the EU's digital strategy, this ambitious technology embodies a new generation of digital devices that do not rely on fossil fuels, equating smart technology with environmental remediation. These breakthroughs would not have happened without the FET funding scheme. Securing 100% finance for risky, innovative projects is essential for bringing about step changes in innovation needed for genuine disruption. The whole experience has been a fantastic training ground for building an ambitious vision, organising complex projects, identifying shared needs, managing budgets, and benefiting from industry expertise. The project-based approach has enabled the consortium to come together around a shared vision, despite our different disciplines, skills, and backgrounds, and make it come true. An unexpected further avenue for applications of living architecture arose during the COVID-19 pandemic. Spotlighting the presence of microbes all around us, even in our homes, the recent lockdown confined us to indoor spaces, where even before the crisis, we usually spend around 90% of our time. Now that we're spending even more of our time indoors, we can think beyond the biology of the virus and into the complex systems that shape the spread of the pandemic. Specifically, we can consider how transmission chains can be interrupted within our domestic environments, where we are most likely to contract the virus, by regulating the overall microbial health of indoor spaces through the installation of living architecture. Currently, cities like New Haven, Connecticut, are using sewers as COVID-19 early warning systems, or canaries in the coal mine, to track the levels of the novel coronavirus circulating inside their populations. If someone is affected with the virus, it shows up in their feces even before they may feel sick. Virus-flecked feces make their way through sewage systems, and checking in on all that sewage gives public health officials another layer of data on the extent of the outbreak. Fed with waste from our bodies, our natural microbiota and any pathogens will enter the living architecture bioprocessor system as we use it. A particularly interesting observation, which has been explored since an early stage in the development of the Living Architecture project, specifically in relationship to removing synthetic organisms, is the ability of the microbial fuel cells to kill disease-causing pathogens in their feedstock. Demonstrated by the University of the West of England, who is currently looking for COVID-19 samples to trial along with hepatitis B and other pathogens, early findings are not only a novel breakthrough for the sanitation sector, but also creates opportunities for regulating the microbiome of the built environment, like an external immune system. So at some point, our microbiota will reach the microbial fuel cells, where through microbial action, they are either incorporated into the anaerobic biofilm or removed from the system. Once a person's or community's microbiota and biomolecules have entered the living architecture system, they can be used for health monitoring more generally. With its integrated systems of artificial intelligence, sensors, microbial actors, and robotic actuators, living architecture provides a programmable infrastructure that can help us rethink how we manage our individual and collective health within indoor spaces to attenuate, for example, outbreaks of infectious disease. Providing a platform for the early detection, localization, and even removal of pathogenic agents from the indoor environment, a near future scenario within a building is through the installation of living architecture. On the detection of pathogens in the system, artificial intelligence and robotic actuators will activate air conditioners and operate an early warning system that, depending on the organism and context, acts according to an institutionally determined protocol. While living architecture won't solve the immediate COVID-19 crisis in its current form, Unfortunately, as COVID-19 has rarely been found in urine, it does alert us to the near future role of the productive actions of microbes within the home environment and even workplaces that enable new forms of economy, health, medicine, public health, and disease prevention, all while making clean energy. Of course, these latter scenarios are speculative right now, but future outbreaks of infectious disease will worsen with a climate emergency and will not be won through an exclusively biological approach that only focuses on a specific organism. As we have already witnessed, our behaviours are also critical in our health and well-being. Living architecture helps us rethink how we make better use of our living spaces, where we spend so much of our time. And even in its current proof of principle form, this combined utilities platform is an important innovation space. A foundational platform and a way of thinking about our relationships with microbes and resource consumption at a critical time in our history. Living architecture genuinely offers an alternative approach than resource consumption and industrial fossil fuel-based systems. It can even change our values, our environmental impacts, and economies. Working with live organisms as an extension of their existing co-inhabitation with us creates the possibility of a new relationship with nature that is not a panacea, but a different way of acting, thinking about, and valuing our world. Not all roads to innovation are direct, efficient, or linear. Not all outcomes are immediately profitable with respect to the dominant economic model, but innovation itself can be empowering and help us combat challenges in new ways. Something occurs during an ambitious FET project, something connected to perhaps the most important things that ambitious collaborations can offer: a commitment to shaping an uncertain future, an investment in those who will be shaping that future, and the formation of community through an expression of trust and sharing that makes all the difference.
Fish AI, Artificial Intestine for the Sustainable Farming of Healthy Fish.
I'm Fulvio Gandolfi, Professor of Anatomy and Cell Biology at the Department of Agricultural and Environmental Science at the University of Milan. I'm the coordinator of the Fish AI project. Nutrition is a key aspect to improve the quality and reduce the environmental impact of producing enough food for an ever-increasing population. Our project aims to develop an in vitro platform that mimics the complex function of the intestinal epithelium. Thanks to the bottom-up creative approach of the FET program, we combine experts in biomaterials, stem cell biology, and fish nutrition with a global leader in animal feed production.
The development of new fish feeds is an ever-changing and diverse global market. Aquaculture production will need to more than double between now and 2050 in order to meet the food requirements of the growing global population. But at the same time, we need to reduce pressure on the planet's resources. Skretting's mission is to be part of this change and to feed the future in a sustainable way. Traditionally, aquaculture feeds have contained fish meal and fish oil as the primary ingredients. But after extensive research into new raw materials, which have led us down some exciting new paths, we are moving towards becoming independent of these finite marine ingredients. This has opened a world of opportunities to use alternative, innovative ingredients, such as microbial proteins and insect meals. But getting here has been a long process. The development of an innovative tool such as the Fish AI could be instrumental in enhancing our progress with screening new ingredients, predicting and optimizing the efficacy of our feeds, and enabling us to develop diets more readily when the needs or challenges arise.
Fish AI, an innovative in vitro 3D culture platform which combines novel fish feeds with a realistic artificial 3D intestine model. What are the essential components of an in vitro 3D intestinal platform? We need fish intestinal cells to create the artificial organ. However, without a suitable support, the cells cannot form a functioning intestine. This is where polymers come into play. A 3D, intestine-like supporting scaffold based on biomaterials is therefore essential. The more realistic the 3D structure is, the more efficient the model will be. In order to mimic the intestine, 3D printing techniques are being exploited. What does the digesta contain? The exact contents of the digesta are currently still studied by molecular biologists. By combining the right components, an artificial intestine can be developed. By maintaining the flow of the digesta through the cell-seated 3D polymeric scaffold, real-life organ behavior can be simulated and studied.
At Biofabrics, we are experts in biofabrication and in developing personalized cell and tissue culture devices. Project FISH AI is enabling us to find new applications for our technologies in animal and nutritional research. This project is also enabling us to find a new, wider pool of potential clients and establishing new research collaborations.
Alternative fish feeds can be tested directly using the in vitro model. After the calibration of the FISH AI device based on feeding trials, the most promising compositions of the novel fish feeds can be sorted out in a time and cost-effective way. It has the potential to make fish meat available to everyone. Isn't it worth it? FISH AI. We believe in a sustainable future.
Honeybees are important actors in the ecosystems we all are depending on. They ensure the prospering of plants and, in consequence, also the well-being of animals due to the enormous amount of daily pollination flights. With the project HIVEopolis, natural bees take a significant step into the age of digitalization, as implementing modern technology into the hive could prevent the species from extinction, even in a century of ecosystem collapses. In HIVEopolis, scientists gathered to design the beehive of the future. The team started to develop a novel type of hive that allows the integration of modern technology and which is also accepted by the bees, starting by studying the design principles from nature itself. Not only the hive had to be reinvented, but also a novel experimental environment had to be built. The HIVEopolis Field Laboratory in the Botanical Gardens in Graz, Austria. It will host many of the HIVEopolis experimental setups and scientific technologies. During the first project year of HIVEopolis, several hive prototypes were produced, exploring innovative materials and fabrication procedures. The important question was which material to choose, as it had to be approved by the bees. In parallel, the material must also allow to integrate and embed all required mechatronic equipment into the hive. The scientists built several prototypes, some built from wood, investigating different space topologies, given by composing the hive from different compartments and modules, which is an important aspect for commercial beekeeping. The final HIVEopolis hive will be made out of sustainable materials and will have a tree trunk-like shape. The project team has followed a paradigm of generative design, that is a principle in which computer algorithms produce the shape of the hive, which is then finally 3D printed and which builds the substrate for additional organic materials to grow. Therefore, different organic materials were developed and evaluated. Finally, a special fungus grown on natural waste was chosen to fill the cavities, which were 3D printed from wooden substrates. This Hiveopolis hive resembles the natural habitats of honeybees, for example a large cavity in a hollow tree, while it is simultaneously enhanced by mechatronics to elevate honeybees in the age of digitalization. Being able to modulate the bee's behavior is one of the key outcomes of the project. One of the methodologies to do so is the Hive's revolutionary traffic control system. This allows users to guide the bees within the hive to specific compartments, which makes bee-friendly hive manipulations possible. In addition, these traffic flows are important for assessing the colony's health and well-being. Influencing the colony's behavior is one of the project's goals. Thus, the project team has developed a methodology they call ecosystem hacking, as it allows to regulate the location, the timing, and the intensity of the pollination service which the bees provide. This way, they can be protected from flying to foraging sites that are potentially dangerous for them. In order to do so, the researchers have developed a set of methodologies to influence the bee dances. Bees use these dances to recruit other foragers to specific food sources. A novel robotic device looking like a robot bee is being developed to imitate the bee's dances. It will be integrated within the beehive's so-called dance floor module to stimulate the natural bees to follow its advices. Secondly, the bee's behavior will be influenced by suppressing recruitment dances, for instance through specially composed vibration signals that stop bees from moving, thus also from performing recruitment dances. For monitoring the hive, the team also invented a novel camera device to be integrated into a conventional honeybee comb frame. This camera device provides an inside view of the honeybee colony's life, comb development, brood development, and of course the bees themselves can be observed. A new opportunity for traditional beekeepers as the device fits into conventional hives. For the final Hiveopolis hive, the project team has started to develop a set of cameras monitoring the hive from different angles. Additionally, experiments to integrate various sensors, like heat and humidity sensors, have been developed, and prototypes of actuators for vibration, airflows or temperature have been investigated to be later integrated into the Hiveopolis hive. A concept the researchers named organismic augmentation. Data plays an important role in Hiveopolis. On the one hand, environmental data is collected and served to the hive. On the other hand, large amounts of data are also collected inside of the hive. Embedded mathematical models have been developed in order to validate and to understand the data and to ultimately support the collective decision-making of the honeybees in a way that strengthens the colony and their well-being, what ultimately boosts ecosystems by pollination flights. The Hiveopolis team was also active in sharing its novel approach and first accomplishments within the scientific community and with the broader public, as several events like the Vienna Design Week were visited. Design and aesthetics are an important key factor for the project, as the new technology has also to be aesthetically pleasing to people while being sustainable in parallel in order to create its own market niches. The project addressed the maker community at specific events in Copenhagen and Vienna, focusing on disseminating its innovative material and design methodologies to this community. Besides these activities, the project team has developed a novel community hive to allow showcasing parts of the Hiveopolis technologies to beekeepers and to bee breeders, which were also addressed through presentations and lectures. Exploitation options of innovative Hiveopolis technologies were explored with various stakeholder groups. More challenging years lay ahead of the international Hiveopolis team, as the goal is set high, developing the beehive of the future.
Our project is looking at how we can use visionary science to transform domestic resources into a broader range of products for the home by using the incredible processing power or metabolisms of microbes. And this fits into an overarching vision of urban sustainability and circular economy, placing the home at the heart of transformation. The steps that we need to make in order to achieve this is a series of developments. So we need to make bioreactors or bioprocessors, which are tiny organs inside our buildings that can process waste and then make useful substances. So we need to design that hardware. To control that hardware, we also need low power electronics. And that is something that we're looking to develop further innovation in. And the other thing is the bio-digital interface. In other words, ways of speaking to and recognizing our microbes so that we can take care of them and they can take care of us.
Global energy demand is growing at its fastest pace, aggravating the energy crisis and climate change. Renewable energy, such as the sun, wind, and geothermal, is clearly the key to our future. But there is an additional and untapped energy source: the heat. In fact, we produce and waste heat through all levels of human activity. If we can convert even a small fraction of this waste heat into, say, electricity, that's a huge savings on global energy consumption. But how can we turn heat into electricity? One such solution is found in thermoelectric materials.
All materials possess the ability to convert heat into electricity through a process called thermoelectricity. When you heat one side of a material, the heat forces electrons to move, turning a part of this heat into electric energy. How efficiently heat is converted into electricity depends on the material itself, and for solids, the best ones today are nanostructured semiconductors. So, thermoelectric materials are portable, renewable energy source. For example, they can use your body heat to power a wristwatch. But the problem with these materials is that they can only deliver small power. They are also made of rare and toxic materials like bismuth and tellurium, and are quite expensive to produce. So we certainly want to make them more efficient, environmentally friendly and cheaper. And that's what the Project Magenta is all about.
In Project Magenta, we use a special kind of liquids called ferrofluids as a thermoelectric material. These ferrofluids are made with magnetic nanoparticles dispersed in ionic liquids. They are highly conducting and also magnetic. Their boiling temperature is above 300 or even 400 degrees Celsius, great for waste heat applications. Through screening and testing, Magenta has now succeeded in producing brand new ferrofluids with promising thermoelectric properties. Thermoelectric voltage and power are generated using a device known as thermocell. A thermocell can be placed near a heat source such as the exhaust pipe next to the car engine or an electronic appliance. Here, we are using a prototype module to test the performance of liquid-based thermoelectric technology for waste heat recovery. Magenta is developing smaller modules designed for recovering heat from small electronic appliances or even a human body. Magenta is developing an alternative thermoelectric technology by gathering researchers from multiple scientific domains: physical chemistry, electrochemistry, physics, as well as electrical and materials science engineering. Together with industrial partners such as Fiat and Solvionic, we hope to build a brand new thermoelectric technology for a sustainable future.
Our institute and other research institutes in Europe are really concerned about the problem that society is facing nowadays, that it's related to environmental issues. The heavy use of fossil fuels worldwide is changing our atmosphere, so we should start some plans to stop this process. One way to face this problem would be to substitute fossil fuels by other type of fuels, energy vectors that are environmentally neutral. And one of these possibilities would arise from artificial photosynthesis.
And this is what the HIVE project is about, to convert solar energy into solar fuels. And for that, we needed to make a device. And we needed the input from all different expertise. So it's a really interdisciplinary project where we work together with people from chemistry, from physics, from material science, from engineering. And we got 8 million in funding from the European Commission to produce the prototype. So it has been already almost 4 years that we are collaborating, and we have the prototype almost ready for implementation.
In the HIVE prototype, the main idea that we like to have is the transformation of CO2 into useful chemicals, for instance, formic acid, or CO, or alkanes. And for that reason, we need to use catalysts that they are specific in order to do this transformation in a selective manner. So we need a prototype where we have different compartments where in each compartment happens one type of reaction. The first one that we find in the cell is the chamber where the CO2 is going to enter and exit, and it's going to encounter our catalyst. That's going to be located in this GDL, gas diffusion layer. Okay. Then we're going to have a membrane, and here we're going to have a liquid where the liquid products are going to be collected. Then we have another compartment which is separate also with another membrane that is not here in this device, but it's going to separate the compartment where the CO2 is transformed and the compartment where water oxidation occurs. Water oxidation is really important because it's the reaction we need to do in order to extract the electrons that we're going to use in order to do the CO2 reduction. And the catalysts that we're going to use in this prototype are based on iron and nickel, so it's really abundant materials. Then the device is closed, and on the top what we have is a photovoltaic cell that is going to collect the photons of the light, give us a redox potential that we are going to use in order to do those transformations. The interesting thing from this device also is that it is modular, so we can change the different parts, and it's easy, so we can easily upscale, but also allow us to study scientifically how this system works and how those components they operate all together.
It is not only a scientific challenge, we need to be able to do this process in such a way that it can actually compete with current fossil fuels prices. Otherwise, it will be very difficult to transform society and substitute fossil fuels if the price is not competitive with current technologies.
ALIF is also an opportunity for the PhD students and for postdocs, for a new generation of scientists, being trained in this interdisciplinary environment and being aware of the problem of sustainability. So I hope in the future, the generation of scientists that we are training will also have possibilities to develop these projects further.
Projects like ALIF, prototypes like the one we are going to introduce in a few months are just starting points in a long road to solve this crucial issue of fossil fuels for society and for environment. But projects like this one are fundamental if we want to find a real solution, a viable solution in the future.
We're bringing a new technology that can revolutionize the way we build our homes for the future.
We are in charge of designing metabolic pathways.
This is the first time in which we are applying biotechnology-based solution in architecture.
We could simulate different external conditions that the bacteria is exposed to.
We work with nature, not against it.
It will be the attempt of incorporating different innovations from diverse scientific fields.
And the interior climate is improved.
Living architecture changes the relationship between home and environment. Instead of something that is depleting and polluting our world, it is actually reconstituting and adding to it.
Humankind is facing incredible challenges, including pollution, global warming, natural resource depletion, waste disposal, loss of biodiversity, and overpopulation. The way we work and live is detrimental to our homes, cities, and the environment. We have to radically change the way we inhabit our planet.
Living architecture addresses the global challenges like climate change by inverting the current relationship between buildings and environment. Right now, we assume that our buildings are going to damage the environment in some way. We actually have to have buildings that promote environmental enrichment, regeneration, even heal and clean the surrounding spaces in which we inhabit. And living architecture is setting out to do that by engaging the processes of organisms, which is very different to the impacts that we can achieve with machines.
It is a completely novel approach that microorganisms are integrated in a building to support the supply systems of a building, to generate electricity, clean water, and recover other valuable components from waste, which is produced in the building.
The concept of the Living Architecture project is to use the environmentally reconstituting abilities of organisms to help our homes become good for the environment.
To develop the project Living Architecture, 6 partners came together. The University of Newcastle as coordinating partner, the Bristol Bioenergy Centre at the University of the West of England, the CSIC Spanish National Research Council, Liquifer Systems Group, Explora Biotech, and the University of Trento. In the Living Architecture system, organic waste and water is fed into the system at one end and is processed through a series of smaller bioreactor units to produce clean water, electricity, biomass, and phosphate. It consists of 3 types of integrated and codependent bioreactors: a microbial fuel cell, synthetic microbial consortia, and photobioreactors. The generation of electricity and the removal of organic matter from the water is the role of the microbial fuel cell.
So the microbial fuel cell has got two half cells, the positive cathode and the negative anode. We have bacteria in the negative anode and algae or just open to air for the positive cathode. The bacteria in the anode consume the chemical organic matter, which is their fuel for growth and maintenance, and they excrete electrons or electroactive byproducts, which then reach the electrode surface, which is inside the chamber. Electrons travel along the electrode surface through a wire to the cathode, where they react with protons and oxygen and close the circuit. And we have looked at how well that microbial fuel cell can clean the wastewater, generate clean catholyte, which is a disinfectant that comes as a byproduct, and produce good levels of electricity. For me, that's the breakthrough. The microbial fuel cell has been integrated in the living architecture wall as a redesigned brick, which has got the anodic chamber for the bacteria. It has got the cathodic chambers for the algae, but it also has additional chambers for the genetically modified organisms. It comes together as a single unit, and we have several of these.
The part of the living architecture system which retrieves the phosphate and polishes the water are the synthetic microbial consortia.
Synthetic microbial consortia is a new to nature microbial community when microorganisms that never had lived together are largely engineered to not only live and grow together, but also to perform a very complex biological function that cannot be done by a single microorganism. The overall concept of living architecture synthetic microbial consortia is based in two different living models. So the farm model that is composed by cyanobacteria that produce sucrose using CO2 as sunlight. With this sucrose, we're going to feed the other module, the labor module, that is composed by two very well-known bacterial workhorses that produce the functionality of the consortia.
This setup makes it possible to engineer several unrelated biological functions, such as phosphate retrieval, within one single bioprocess. To design synthetic microbial consortia, the team has used experimental and computational methods. A computer simulation has been established to model the phosphate metabolism in E. coli bacteria.
And these simulation results are then compared with experimental results. And then we interrogate the models to provide response to our problems, to understand, for example, how E. coli will respond to different environmental situations. And this way we can understand how we can tune the bacteria by using synthetic promoters so that it will provide the right response that we would like to integrate into the biotechnology, which is going to be developed.
The first thing we learn is that we have to provide researchers with effective tools to engineer complex pathway. And together with the University of Trento, we completely redesigned our computer-assisted design software. And we now are capable to deliver an effective tool to tackle biological complexity. Dulix brings together data, softwares, and reagents to allow people to work faster, better, and safer to deliver the synthetic biology revolution.
Oxygen is brought to the system by the algae in the photobioreactors.
The role of the photobioreactor is a little bit like the lungs of the system. It's delivering oxygen into the system when it needs to breathe. It also reminds us that the algae and the plants on this planet are the lungs, and we rely on them to live anyway. So we want this to be as low energy as possible. The best way to do it? Let gravity do the job for you. The liquid comes down the top, gravity very, very slowly draws it down, and it does the work for us. We're using nature. We're not working against it, we're working with nature. The uniqueness is that it combines functionality, practicality, but with an artistic, aesthetic element to it. It's pleasing and it works at the same time.
The prototype built by Living Architecture Consortium is controlled by a custom-built electronic circuit, which ultimate goal is to be an autonomous system in terms of electrical energy. The system is comprised by an energy harvester, which extracts energy from the wall components, and low-power microcontrollers and peripherals that enable the system to be autonomous in terms of energy.
The most significant results of the Living Architecture project are really using household waste as a substrate for producing energy, recycling water, and even making useful substances.
A wide range of scenarios for applying the Living Architecture system in our homes have been developed.
The applications are reflected in our use cases, which we developed for retrofitting of existing buildings, but also as design concepts for new buildings, for single-family homes, for multi-family homes, for office buildings, and for school buildings. And in addition, we developed a concept design for a multifunctional building in a very dense urban environment. The bioreactor system covers the whole building. It is fully automated and it can be accessed from the general circulation area. And we integrated a vertical garden for combined socio-ecological impact. The vertical garden enhances the system performance, and the interior climate is improved. And it offers an interesting place for people to meet, so it promotes social cohesion.
Living Architecture is a step towards a circular economy. The project demonstrates that organic waste can be separated within the building and transformed on-site into valuable resources. Living Architecture pioneers the transformation of buildings into regenerative systems. It uses biology to solve critical environmental issues for sustainable and livable cities in the future.
In HOT, European universities are working together with the industrial giants IBM, ST Microelectronics, Hitachi, and Thales SA. The Hybrid Optomechanical Technologies project brings together 17 research groups in 11 countries. They have demonstrated proof-of-concept devices with potential market readiness in the near future. But what are HOT devices? Modern communication runs on optical fibers. We depend on light to catch up with friends and watch cat videos. Unlike in sci-fi movies, crossing beams of light achieves nothing, so we can send lots of light on one optical fiber. However, light can push tiny mirrors, like wind on a sail. This radiation pressure allows light to interact with itself. A beam of light hitting a mirror moves it. The direction of a second beam then changes depending on where the mirror is. By interacting with a moving object, the first beam has affected the second. This moving object acts as the medium that gives rise to an effective interaction between the two light beams. This brings us to the optical circulator, which acts as a roundabout for light. HOT's circulator uses a microscopic silica toroid in which light can circulate 100,000 times. Just like we saw earlier, light transmitted through this device can be controlled by another light beam with the help of mechanical motion. This control beam induces unusual behavior. When light carrying a signal enters the device, it can be transmitted directly or converted to mechanical vibrations and back to light. These two pathways can interfere constructively or destructively. As a result, light entering port 1 exits through port 2, where the interference is constructive, and not port 4, where it is destructive. Light entering port 2 is routed to port 3 instead of 1. This circulator is a non-reciprocal device. Since light can go from port 1 to 2, but not vice versa, it is unique because it does not use magnets and is controlled using light itself. Hybrid optomechanical technologies have several other applications. Over the coming years, HOT will be investigating hybrid sensing devices as well as enhanced positioning systems that are ideally suited for mining and navigating deep underwater. These devices will revolutionize the internet, making it run more efficiently. Light is also an ideal carrier for quantum information, so these devices, as well as other devices under development by HOT, can pave the way for a quantum internet.
Good afternoon and welcome to this Future Tech Week session entitled A Female Perspective on Robotics and Information Technology. My name is Sarah Keane and I, along with my colleague Erich Prem from OPTOMECHANICAL, we are members of the FET-FX Consortium. Today we are speaking with two impressive women from EIC Pathfinder FET projects. Professor Cecilia Lasky from the Biorobotics Institute of the Scuola Superiore Sant'Anna in Pisa was the coordinator of the FET Proactive Project Octopus, which ran from 2009 till 2013, and Professor Clivia Sotomayor Torres from the Catalan Institute of Nanoscience and Technology, who also happens to be a former co-chair of the Future and Emerging Technologies Advisory Group and coordinator of the FET Open Project Phenomenon, which ran from 2016 until this year. Welcome to you both, and thank you for joining us. So I will launch right into our questions then. I'll start with Professor Lasky. Perhaps you can give us a brief description of the Octopus project, highlighting some of the challenges it addressed, the results and the impacts. Also, have you been involved in any other notable FET projects?
Octopus is really my heart because it was a very interesting project for me. I'm a roboticist, and at that time I decided together with colleagues to study the octopus as a model for robotics, and that was quite unusual at that time because an octopus is completely soft, so completely different from a traditional robot. And then the challenges were so many because we had to learn a lot about the biology of the animal. And then we had to translate the principles that we discovered, and that part was very, very interesting. But those interesting principles had to be transferred to engineers. So we had to develop the technologies for building a robot that was soft, that could contract, that could elongate an arm and things, strange things like those. But actually, it worked a bit. We demonstrated at the end of the project that it was possible. We built a few prototypes, and especially we developed a lot of knowledge and new technologies that were at the base of what we call today soft robotics. So the impact in this sense was enormous because today there is a large community, a large scientific community around this field. Robotics that for me it was very much nurtured by the Octopus project.
Great, that's really interesting. And Professor Sotomayor Torres, please tell us a little bit about Phenomen as well as any other FET projects of note that you've been involved with.
Okay, Phenomen was a journey of discovery in many ways. I am passionate about the physics of lattice vibrations or phonons, and for some time I've been harboring the hope that we could use phonons for information technology in general, particularly information processing. And I met these like-minded people, and we decided to say, right, let's build a circuit, but not one that works at 10 millikelvin, one that works at room temperature. And not that one that works with poisonous materials or expensive materials, but straight silicon. And these were the boundary conditions that we set. We generated various concepts for particular enhanced functions that would give us properties that could be harnessed into a device. So in this project, we managed to build, design, set up, and demonstrate a phonon source, a phonon detector, a phonon switch, and all was based on the interaction of phonons and light. Phonons are rather elusive, although they are ubiquitous, they are everywhere. And what was very important for us was to have an interaction like that between lattice vibrations and light, electromagnetic radiation, that would allow us to understand what was going on and point us in the direction of building a circuit with minimum losses and that could perform information and communication technology functions. And thanks to an extension of 6 months, we managed to do that just in the last 4 weeks of the project. So
just in time.
Perfect timing. So we recorded the video with the experimental results as they were coming out of the lab. And we received the news that we had lockdown in Spain from the next week. So it was, oh God, just everything on time somehow. But it was fascinating. And what really was very good and very encouraging and gave me a lot of hope in the future is the commitment of the young researchers. It was amazing. They were involved day in, day out. They were prepared to take extra work and participate in events like the ICT in 2018. They made beautiful graphics, they prepared presentations. They had the courage to present their work in front of the reviewers that we had for our project. I said, no, of course you can. And this is part of our job. It's also not only to push the science, but also to train the next generation as well. And this experience I have also had in other projects. Before Phenomen, I coordinated a project of fed energy that was called Merging, and it was based on membranes, on silicon membranes for energy harvesting, which actually laid the foundations for Phenomen in many ways. Before that and other projects, but I think Merging was a stepping stone towards Phenomen. And Phenomen is a stepping stone for other things now.
That's great. Okay, so tell me a little bit, like, how would you describe the innovation potential from Phenomen? The project has very recently ended, so are there some new projects that were going to be related that are on the horizon, or some other transition to innovation activities that are planned?
I think our consortium was very aware of the FED Launchpad, and so in particular one partner who is very oriented towards devices for technology found a match in an SME, and they decided to apply for a FED Launchpad project, which they got. And so they will be working on oscillators to read radio frequency signals based on the radio frequency coupling we use in Phenomen. So that is one success story, I would say. There is a business plan, it's all very clear. I think they have a very high probability of success there. And obviously, there were many questions left open, and they were worked into proposals that were submitted in the last round. So there are 2 or 3 proposals that came out of Phenomen by different consortium members with other consortium members or with other new partners, which is perfectly fine. So
that's great.
So I'm very happy with that, yes.
Great. So Professor Lasky, Octopus ended in 2013. Can you tell us a little bit about some of the developments that have happened since its conclusion?
Sure. I mean, I agree with Clelia a lot about the training of young scientists in this project, which is really, I think, the strongest value of our results. In terms of innovation, I was not as lucky as Clelia, because the Launchpad was not there yet at that time. So I couldn't apply for that. And actually, I found a gap between the results from such, how can I say, basic research, fundamental research project like an octopus. So we ended up after 4.5 years with a prototype of an octopus-like robot, but it was really a demonstration that it was possible to use software. It was, I mean, just a demonstration that it was possible. We could investigate more about soft materials in robotics, but from there to other kind of projects where you have to address some application or you have to integrate a real system, there is a gap. So we tried to submit proposals, but they weren't accepted because it was too early for that. Fortunately, actually, we could go on with some funding, and we went on developing our technologies, and the field grew, so many technologies became available all over the world. And suddenly we found an interest directly from companies. Companies interested in applying our let's say octopus-like robots, but with the time, along the time, it was no more octopus-like. I mean, the important is the technology, the soft robotics technology. So the soft actuators, this kind of robots that can stretch, that can change shape, that can be soft in the interaction with people. And we found the interest of some companies, and we had also directly some funding from companies. Of course, the field of applications for a soft robot are so many. The biomedical field is one of them. You can think of soft arms assisting people. And there we had a project funded by the European Commission. The same in surgery, endoscopy. In endoscopy is very useful to have something soft that can move. So we also find interesting applications underwater, because we learned a lot from the octopus about locomotion and manipulation underwater. And also in the oil and gas companies, for example, so what is related to exploration, monitoring, and last but not least, we are now finding a lot of interest in those people and company monitoring the sea bottom. So it can be for the industry, it can be for ecology, can be for biology, can be for climatology, oceanography. So really many applications are coming up today, and the same for the single technology.
Thanks to both of you. I think this is really interesting to see how many different applications and you're mentioning, and how many areas this is opening. So I'm sure you are aware of the Commission priorities, you know, ranging from the Green Deal to the digital age, and Europe's fitness for the digital age, and others. May I ask you, what's the relation of your research to these priorities, if any? And maybe Professor Lasky, if we can start with you.
Yes, I started from the last thing I mentioned, that is the application of our soft robots underwater. There, I think we can contribute a bit to the Green Deal priority, because in two ways, actually. One way is what I mentioned, so we can build robots that can help in monitoring the environment. Because we can help robots that can move autonomously in different environments, even underwater. And that's one part of the story. So we can, provide them with sensors. They can measure a lot of parameters that are of interest for ecology, for the environment. But on the other side, using different materials for building our robots, we are also investigating materials that are more compatible with the environment, so eco-compatible or even biodegradable material, especially when we work at sea. I mean, we realized very soon that we couldn't put in the water of the ocean a robot which is made of materials that are not completely compatible. They can be dangerous for animals if there are dispersion of substances. So that's something that we are feeling very strongly today. And so we started a new research in our group about biodegradable materials that can be used for marine robots, so that we can even think of robots that can be sort of disposable at sea without hurting the environment. And I think that this is a strong concept that if it is used also in other fields of robotics, but also in other fields of technology, just think of the digital technology, the smartphones that we are producing and wasting today. If you can use something, some materials that are more compatible with the environment, that would have an enormous impact. And since I work on robotics, so I build robots, of course, my research also relates to other priorities that concern more in general the economics related to the jobs and occupation. Of course, when I say the word robot, the first question I receive is that, okay, but are you stealing jobs in your What I personally think is that robotics is instead can be very great source of occupation, especially in Europe, because in Europe, I mean, we are quite strong in the robotics industry. So, but again, we are thinking of a new generation of robots. So I think that what we should do with these new materials, with these new abilities that our robots have to go and explore environment, to access difficult environments, is not for replacing humans in jobs that I mean, bring them salaries, but replacing humans in those jobs that are inhumane, if you want, and there are many of them. So again, robots that are more compatible with the environment, that are more able to access the environment, to be autonomous there, and even more compatible with society and with our society and with our economy, the economic model that we have today. And I think it is possible with this new generation of robots.
Thank you, Professor Sotomayor. I guess the priorities of the Commission also go nicely with your project, don't they?
Of course. For example, in the Green Deal, there is a lot of emphasis on the efficient use of energy and also in perhaps even reducing the amount of energy that we use. And the information carriers that we are proposing, the phones, are in fact one of the lowest energy information carriers. And in that sense, there is a potential there to minimize or to reduce the energy consumption in the ICT sector. Not in 5 years. I mean, I think we're looking at like 15 or something like that. At the same time, there is also the prospect of using this knowledge to harvest energy using phonons via thermoelectricity, via vibration, via piezoelectricity even, mediated by phonons. So in that sense, we have, I think, the underlying knowledge is being structured to go towards a solid development and support the Green Deal. Concerning the digital age, I think many of the aspects that are being pursued, 5G and 6G, and etc., they will require autonomous systems. And autonomous systems need not only to work on their own, but also to feed themselves with energy on their own. And there again, this energy factor is going to play a significant role. The edge computing devices also need to be in many ways autonomous. That's the whole idea, is to decouple them from the centralized cloud in many ways. So again, using phonons interacting with electrons, phonons interacting with photons, phonons interacting with magnons, that would enhance the palette of resources, technological resources, to support the digital age as well.
Right. Maybe Professor Sotomayor, because you alluded to it a little bit, you mentioned this long-term nature of FET, and you know, of course, that FET is a special funding scheme. It has been around for 30 years. It's research beyond what is known. But it has a technological objective, even if it's long-term, so it's kind of special. I mean, what are the characteristics of this scheme that are important for your research? And what are some features of the, let's assume, a fully fledged EIC Pathfinder initiative that you feel positively about?
I think what we've learned in FET over all these decades has been that the value of consortium-based research with a layer of multidisciplinarity, with a layer of various generations, the senior professors down to the master students, is an incredibly rich pool of ideas, practical ideas, implementations. It is something you do not find just in a single research group, unless you run a group of 50 people perhaps, and all with different skills. But then again, it is important to have deep knowledge in your area that you can share with people who have deep knowledge in their areas, because that's the only way, in a sense, to make progress, solid progress, sustainable progress that will yield something more imaginative, more useful, and in the future. So for me, those characteristics of consortium-based, which basically means, according to the conditions of the European projects, has meant 3 teams from at least 2 or 3 countries. I never remember the numbers exactly, you always have to go back, but it requires this different way of doing things, because the diversity in Europe is incredibly rich in that respect. The different ways of arriving at a conclusion or formulating a question, and that alone, I think, as a mechanism, is extremely, extremely important. Yeah. So for me, FET remains the most valuable instrument that my generation has had over the last 30 years. And I think it will be important to transfer some of these principles, these operational principles, to the future or the actual European Innovation Council. I have a lot of hope in the European Innovation Council. I think it's a new facet, but I think that research at TRL1 level must be done also in a consortium, not only by solo researchers like in the years.
Thanks. I'm sure Professor Lasky, you have an idea about that as well.
Yes, exactly. I fully agree with Clivia. What I really love in FET is that it's the place where you can really start completely new ideas, new research field. And when I say completely new, I mean completely new, because many other programs claim that they fund basic research, fundamental research. Okay, that's one thing. But in fact, you can really try and submit a completely crazy idea where you don't have any expertise. This is an important point in my opinion, because sometimes, as I said, also compared to other programs, Of course, the CV of the proposer also counts, but in that way, you always bring back the research to the expertise that is already there. So if I must be credible in what I propose, it means that I'm going to do something incremental with respect to my own expertise. The octopus is a perfect example for me, because before octopus, my research was in humanoid robotics. And humanoid robots are rigid, they have joints, you can count them, and you have a number of degrees of freedom. And of course, it can be more or less complex to control them, but that's the story. When I proposed the octopus, I didn't know anything about the animal. And I didn't know anything about materials. I didn't know anything about the soft actuators. Of course, I had started some preliminary studies by myself in my group. I had contacted the biologist, as Clivia mentioned. That's a very valuable part of the story, because I could match with all these people. It required a lot of time to prepare the project, because it is a way to start the research, connecting with the right people, learning the important things to start. And when I proposed the octopus, you know, I sent this project and I said, okay, it's a completely crazy idea. I'm not credible because I'm a roboticist and a computer scientist. So either they will look at it and throw it away laughing, or they will fund it. And unfortunately, it was the second. Actually, this risk that FET took at that time was very rewarding, because as I mentioned today, there is a wide scientific community working in soft robotics, and everything started with that. And the FET program also funded a coordination action that helped consolidating this community. And it's a pity that, as I mentioned, sometimes there is a gap and you cannot go really much further than this, because Europe invests through FET in this new field with a lot of potential reward, and then should be able to take also the results of that, the fruits of that.
Great. So Professor Sotomayor-Torres, you were actually the co-chair of the Future and Emerging Technologies Advisory Group. Can you tell us a little bit about your experience in this role?
Yes. First of all, it was a really valuable experience. I consider myself privileged for having worked with highly esteemed scientists whose number one priority was emerging technology in Europe. Also working with extremely committed members of the unit, of the FET unit. I think the knowledge one gains, the development of new ideas, of trying to find and identify which topics may be suitable for initiatives. This was a tremendous challenge. I think most of the time we managed to propose something sensible in sense of coherence, because sensible in terms of the knowledge perhaps is still open. I found that the cooperative spirit was tremendous, the level of commitment of everybody. And these feel as a unifying European experience was very important for me, not at the level of implementing a research project, but implementing discussions to go into documents that could be perhaps considered by the Commission when they decide on their policies. I had just the impression that several times there were things that looked like fait accompli, that one could try to perhaps modify a little bit, but the core decisions in terms of core policy, of course, they are made by the Commission. and we try to simply modify it and leave room for basic research, oriented basic research, or translational research, whatever adjective you choose. For me, is all technology readiness level 1. Yeah. One important aspect was the discussions that I learned a lot from concerning the role of the social science and humanities. And for me, this has been now during this pandemic has become so apparent, the need to have this understanding. Yeah, and perhaps this is something we don't do enough. Perhaps there is some reluctance from the social science and humanities. Perhaps we are not doing things in a way that they can be truly involved. Perhaps we need a new generation of social scientists and humanists that can talk to our younger colleagues, because clearly people of my generation, we are pretty set in our ways, even if rationally we want to. We lack the skills or the ideas to approach the social sciences and humanities in a way that they can become truly involved. This pandemic is opening an enormous opportunity as well. To solve many of the problems, we cannot ignore the social science humanities. We have economics, we have psychological issues, we have all sorts of things, organizational, philosophical, and they're all based on a phenomenon, this pandemic, which has, in principle, only a technological solution in the first instance, but for it to be long-lasting, it needs a social science and humanities. So this is something that I also learned to value a lot in the Future Emerging Technology Advisory Group.
Thanks for that. Let's just stay with this question of social aspects a little bit. And there's one thing that I wanted to ask you, although we don't want to focus entirely on it, but obviously we have two outstanding female professors here who work in not just science, but also technology. And on the other hand, technology is often considered a male thing. I mean, Professor Lasky, what are your thoughts on this?
Well, I must admit it is true. It is usually considered as a male thing. But of course, I cannot say why. If we look at numbers, at statistics, so we see that it is quite true. It is true in the industry. It is true in the workplaces. It is even true in students. So in the universities, when female students have to decide what kind of path to choose, we know that there are a few of them that decided for some technological hard scientific field. Of course, I don't have an answer, but I can just bring my personal experience in that. So when I was a student, it was really a male world. But as a professor now, I see that things are changing. And especially as a professor, I am in the biomedical engineering field, and the kind of robotics I do is biorobotics, which means the biomedical application of robotics, but also the study of biological systems, like my studies about the octopus. So there I see a much larger percentage of female students. So in biomedical engineering, we have an equal number of males and females in our classes, and also in our biorobotics institute, we have a very large percentage of female PhD students and scientists. So that's just an observation from my side. It's not a real answer. It's not a real explanation of that. But I don't know, probably we know that a biological field attracts more women. If you go to a biology class, you probably find more female students than male students. So the combination of technology with biology or medicine is probably what attracts more women.
Yeah, let me ask a quick follow-up question then to both of you. I mean, and it's a bit provocative, of course, but do you think that women create different technologies or create technology in a way that's different from how men do it? I don't know, Professor Lasky, and then we switch over.
Well, it's difficult to say again. I don't know if we have a different feeling about technology. My own experience is that, as I mentioned in my group, we try to build robots that are more compatible with the society, more compatible with the environment. So we are quite sensitive to this kind of aspects of robotics, including the social aspects that Clivia mentioned, that I also think are very, very important, especially in robotics. There are so many implications that go towards the social sciences. So I personally have this kind of feeling, this kind of sensitiveness towards those topics. I don't know if it is because I'm a woman or just because I don't know, my personal cultural background, and of course my group has females and males, and we all go towards this kind of objectives. So I don't know if it is my guidance or even the male scientists feel the same. Probably we have a little bit more more sensible to this kind of aspects a little bit, but it's not a real scientific answer. So as a scientist, I don't like to give answers that are not fully demonstrated, and these are not fully demonstrated.
That's entirely fine, Professor Lasky. I don't know, Professor Sotomayor, would you like to add? What's your take on this?
First of all, I really feel represented by many of the comments of Cecilia. I think this issue requires a great deal more study, because it's very difficult to generalize in this case. A lot has to do with a set of values, and many values come from socialization. It also comes and are strongly linked to the level of education and culture that one has received. Yeah. And this question of values then tilts the balance in terms of competition at any cost, right? Or healthy professional competition to see which idea or which technology is better. Yeah. It also has the factor that the funding agencies which are not state-run may use other parameters in the analysis and favor projects which they feel are safer or will be better managed. I think there are so many subtle aspects that would need a serious analysis from the social science and humanities, for example, that could unravel. First of all, I mean, I don't like this reductionism, but I'm afraid I was trained in that manner. So I reduced the problem to economic factors, psychological preferences, lifestyles, ability to plan, resilience, etc., and how this is projected and valued by those who would fund and therefore enable the research and the creation of technology. So I would really welcome a study that goes way beyond what has been seen so far of counting statistics and just looking at how these numbers move along the years. My first engagement in Women into Science and Technology was in Britain in 1983, and then in 1984 I joined the initiative of the Royal Society of London, Women into Science and Engineering. And it was an eye-opening, but you see, I look at some of the publications and listen to the debate nowadays, and it hasn't moved very much. it has not moved in all these years. And so we just see this incremental improvement on participation that will still need 100 or maybe 200 years. Right? And in the meantime, we lose all the value that a different way of thinking, a different socialization could bring to new ideas, innovation, and therefore technology.
Interesting. So I have one final question that actually feeds, I think, from that. Perhaps I'll start with you, Professor Sotomayor-Torres. Many countries are trying to increase the number of female scientists and engineers, but they're actually finding it quite difficult to do this. So for you personally, why weren't you scared to study and enter into this field? And do you have any ideas for how females can be further encouraged to enter the STEM fields?
Well, my mother was a social scientist, and basically she said, whatever you want to do, what's the problem? And I said, well, people say family and career, of course you can do both. And so there was never a question. Do what you want to do what you're happy with, and surely you'll find a way to do it. And it was fantastic. And I just never doubted that it was possible. And there was another reason, this historic. I am Chilean by birth, and when I was making a decision on what to follow at university, it was a time of great political upheaval. And I went into physics because I wanted to help my country to be technologically independent. So there was this other motivation as well. Yeah.
Nice. And how about Professor Latsky, in your case?
Yeah, so in my case, I actually didn't consider this aspect much, I must be honest. So I took many elements into consideration in my choices, of course, but I never considered this one as one of the factors for making a choice about my studies, my future career. I must say that I completely disregarded it, but not because I was so smart to understand that it was not so important, but just because it was my nature. So I never considered it as a possible obstacle, or I never considered that it could have been difficult to work with men. Actually, I never thought about that. I had to do when some other people recommended me to consider that. Oh, but are you sure you're going in an environment with only men, or I mean mostly men? Are you sure? And at that time, oh, okay, yeah, I didn't consider that. But and this is something that it's very natural for me. I mean, again, I'm a person that considers so many factors before making a choice, but never this one, never the gender. The gender factor. So that's also my recommendation to young students, female students. I mean, consider everything you wish, but not this as an obstacle. Or I like very much what Clivia said about this feeling that women have of making a choice between a family and a career, especially in science, and that's completely wrong. I mean, we shouldn't think that we have to make a choice. So again, my recommendation is just follow your attitudes, your interests, your passions, because passion is very, very important in every job, but especially in science, in research, and passion should be the first one. But not gender related.
I would really like to thank you both for an interesting discussion. I mean, it was fantastic. And I wish you all the best for your future research. And yeah, thank you for taking the time to speak with us as part of Future Tech Week. We really appreciate it.
Thank you very much. And I hope we can both be among the many actually role models that are available to the young scientists today.
Absolutely.
Thank you.
Thank you.
Thank you.
Leila is a researcher, and she recently secured funding from the European Commission for a FET EIC Pathfinder project that promises to revolutionize the field of quantum technologies. She is very excited about this, but she doesn't know how to identify the innovation and market potential of her project, nor how to set priorities and dig into the exploitation opportunities of her research. Navigating online, she finds out about FET Briefing, a project that helps researchers recognize and leverage planned and unexpected results for market uptake through a portfolio of ad hoc and free services. On the FET Briefing website, Leila participates in a 2-minute assessment of her project and, shortly after, an expert provides her with a tailored service roadmap. She decides to attend the Interactive Innovation Opportunity Workshop, where she learns which skills she needs for her project's exploitation pathway. Leila also attends a communication course, where she learns how to effectively explain and promote her project's innovation to different stakeholders. During a FET Briefing peer-to-peer exchange event, she meets Carl. He coordinates a FET project at a more advanced stage, and he's considering whether it would be opportune to bring some of his research results to the market. For this purpose, he has already attended a set of dedicated FET Briefing courses. He also suggests Leila accesses the FET marketplace, where she can find matchmaking opportunities and meet other potential business partners. Thanks to their enhanced skills and awareness of their research innovation potential, Leila and Carl attend an online pitch organized by FET Briefing, where Carl gets in touch with a pharma manufacturer interested in investing in his project outcomes. Thanks to the capacity building process provided by FET Briefing, Leila, Carl, and many more FET EIC Pathfinder researchers are empowered to make the leap from lab to market.
My name is Cecilia Lasky. I'm a computer scientist, but I have a PhD in robotics. I'm a professor of biorobotics at Sant'Anna School of Advanced Studies in Pisa. If you think about a robot, how do you picture it from the back of your mind? Perhaps it looks something like this humanoid we have here, which was built with rigid materials such as metals. There are parts in motion that, for example, simulate the movement of our arms or of our legs in this case. But what if we come up with a robot built with something so soft, such that it completely distorts itself, like this arm that is made entirely out of silicone? This is what the octopus has taught us. A few years ago, with the support of the European Commission and the FET program, we explored the possibility of building robots with different materials, with soft materials. By studying the octopus, its principles allowed us to simplify robots' control and sensory motor behavior, therefore enabling them to be more efficient in the real world. When we started the Octopus project, everyone asked me, "What is this for?" Well, it's not the robotic octopus, but the principles that the octopus teaches us that serve some purpose. And indeed, as we were working on the Octopus project, we were able to start other projects that had some applications. Let's take, for example, the project on a variable stiffness endoscope, inspired by the variable stiffness of the octopus arm, or another one on a robotic arm for the personal assistance of the elderly in the shower. But there has been a huge scientific impact as well. Soft robotics has now become a vibrant industry that is still growing worldwide. We started as pioneers. Europe was at the forefront of it all. Now people understand that it is an interesting sector. The Americans and the Asians, such as the Japanese, Koreans, Chinese, are investing on it, of course with a greater economic strength. So Europe will risk losing this competitive edge if it doesn't keep its investment. Anyway, as for me, now that over 10 years have passed, I have already started thinking, I should start thinking, about my next FET idea.
d'affetto.
The next generation of artificial intelligence will be explainable and collaborative. Artificial intelligence is a game changer for a variety of sectors. AI's boost to the global economy by 2030 is forecasted to be $15.7 trillion, source PricewaterhouseCoopers, and by 2021, 80% of emerging technologies will have AI foundations, source Gartner. However, this massive potential will only be realised if humans, whether it is a doctor, a manager or a scientist, are able to trust the outcome of AI models and use their own expertise to validate and improve them. This is virtually impossible with the current black box models that dominate AI. These models achieve great performance but cannot be explained. The next generation of AI will therefore have to increase this explainability. Our approach promotes AI and humans working in concert to find better solutions, models that are both effective as well as comprehensible. Our tool is Trust, a transparent, reliable and unbiased smart tool. By being explainable by design, it will be transparent, reliable and able to prevent undesirable biases. Most of all, Trust will allow humans to gain insight and understand the workings of the AI models. Trust will be demonstrated and validated in 3 use cases. One in healthcare for tumour treatment, one in online retail for selecting delivery time slots, and one in energy for supporting demand forecast at buildings and beyond. Yet the true potential of Trust will go way beyond these trials. It will impact society at large and find applications in a wide range of sectors, as for example banking, insurance, manufacturing and public administration. Trust AI works for a transparent, reliable and unbiased AI which humans can understand and collaborate with.
Will electromagnetism technology last forever? Electrons involved in electronic applications have some limitations. There's a speed limit, and they also heat up components. At Phenomen, we are developing a radically new technology based on phonons that will mean a breakthrough in the fields of informatics, electronics, and telecommunications. An interdisciplinary team, including 3 research institutes, 3 universities, and an industrial partner, is working to lay the foundations of a new information technology. Phenomen is a future and emerging technologies open project designed to harness the potential of combined phononics, photonics, and radio frequency. To generate electronic signals by converting light into vibrations and vice versa as information carriers on chips. Ideas like this can change the world we live in. Phenomen, new information technology.
So this is the experimental setup that we use for doing this electro-optomechanical characterization of the Phenomen samples. So essentially it's an optical setup in which we measure the transmitted light through a detected fiber on an optical detector. And so here you can see the fiber optical circuit. It comes from here. Here is the fiber, it goes here. Here is the taper fiber, okay? And here in the computer we can see an image of the taper fiber where we have made a loop shape in order to be locally able to excite one of our optomechanical crystals. And then the optical fiber circuit goes on through here, and here is where we take the transmitted light. Also we use a circulator, an optical circulator is here, so here is the input, and here we get the reflected signal that comes from the signal that is decoupled from our structures. In addition to that, we have included a pair of electrical tips in order to be able to electrically excite the samples, the contacts that are contacting the IDTs that allow the surface acoustic wave generation in our system. And yeah, we can see in the image in the computer the two metallic tips that are placed very close to the optical fiber. So here we're making an image of the sample as it is. We can see the optomechanical crystal here, and this is the optical waveguide that allows the coupling from the waveguide to the optomechanical crystal. We will put our fiber on this part of the waveguide, which has this tapering that allows the efficient coupling from the optical fiber into the waveguide. Now we will put the focus on the bottom part of the fiber. You can see this is the bottom part of the loop, so it reflects this part. It's making image of the bottom part of the loop. And now I will go close with the sample towards the optical fiber. Here on this plot we are plotting the transmission, the optical transmission at the end of the optical circuit. So we have everything now that the fiber is not in contact with the sample. Everything is transmitted, nothing is reflected, and now I will contact the fiber with the waveguide, and we will see a decreasing in transmission and an increasing on the reflected signal. Indeed, I will go little by little approaching the sample towards the fiber until we make the contact, which is happening now. Okay, so you can see a huge decrease on the transmitted signal and a huge increase on the reflected signal. So we have on the left transmission, where we will not see anything, as in transmission we just see that everything is coupled into the waveguide, and it's on reflection where we see that the reflected signal at some point shows a resonance behavior, which corresponds to the resonance of the optomechanical crystal. Here it is. So you can see that there is a mode at 1595 nanometers that corresponds to an optical resonance of the optomechanical crystal. Okay, so here we can see that we are already exciting the optical mode that we were showing before, and we are in a condition that we are able to transduce optically the thermally activated motion in the gigahertz range. Indeed, here we can see like two peaks, two mechanical modes that are thermally activated. The most intense one happening at 2.058 gigahertz. Okay. We are almost prepared to go into electrical excitation of the IDTs. We have one electrical tip that is already on contact with one of the contacts of the IDT, and the other that is about to be in contact. Okay. Then here we can see a vector analyzer that is already generating an electrical signal, okay? So now we will take the reflected signal coming from the optomechanical crystal, and we will connect this signal to a detector that is connected to the vector analyzer. Okay? Already driving the optical resonance, and we are in a condition of transducing the mechanical motion. And here we are using the vector analyzer, in which port 3 is feeding an electrical signal, so where the mechanical modes that we were showing before in the spectrum analyzer were present. And now we are going to go down with the mechanical tips to contact the IDTs. So I will go down first with one of the tips. So this is already in contact. So already we see some signal appearing, okay? And then I go down with the other electrical tip. Which is going to contact now. As you can see, a huge signal appeared, and for example, where we have this marker, it's 2.056 gigahertz. That we were seeing before in the spectrum analyzer. Also, we have here another peak at 2.0797. So here we went back to the configuration in which we are transducing the mechanical modes in the spectrum analyzer. So here we have our mode that now is at 2.055 gigahertz. Okay. And in the vector analyzer we are generating a CW wave at 2.054 gigahertz, okay, with minus 20 dBm. Now we are not in contact. Get into contact with the mechanical tips. We will go down with the mechanical tips. Once in contact, and now I will carry to go down with the other. And at the moment I will be in contact, I will see a strong peak in the spectrum analyzer, which is what we see already.
The Simple Show explains the graphene flagship. This is Peter, physicist from England. He is experimenting with graphene and other 2D crystals to find new ways to benefit from its unique properties. This is Susan, biologist from the Netherlands. She likes graphene for its biological compatibility and uses it for DNA sequencing. Anna is a chemist and works in Poland. She finds graphene extremely interesting due to its two-dimensional structure and is experimenting on its modifications. Overall, there are thousands of scientists in hundreds of labs across 29 European countries studying this exciting and most promising subject in science nowadays. David, a talented engineer, has a more practical approach to graphene. He works for a big electronics company in Finland. His boss commissioned him to develop new applications for their products. To accomplish this goal, David needs the expertise from scientists like Peter, Susan, and Anna. But where does he meet them? At the Graphene Flagship. This graphene community, initiated by the European Commission, joins both scientists and industries. Thanks to the Graphene Flagship, David can quickly build a network to scientists and hundreds of other European companies working on graphene applications. Due to this collaboration, the entire value chain, from materials production to component fabrication and systems integration, can be covered. In long-term perspective, this means new ideas in our heads, new opportunities for skilled workers, new products on the market, and finally new potential for economic growth in Europe and around the world. With people like Peter, Susan, Anna, and David, the Graphene Flagship is creating the way to our future.
Nanomedicine is getting smarter, improving our ability to treat cancer. The size of nanoparticles makes them ideal vehicles to carry drugs to tumors, and we can modify their surface and material to make them better at moving through the body to reach their targets. Our focus is on treating cancer stem cells, because if we don't get this right, then treatments might not tackle them, and these cancer stem cells will then lead to the recurrence of the tumor.
The goal is to transport therapeutic agents by using nanoparticles as carriers deep into a tumor in order to reach all cancer cells. However, most of those nanoparticles accumulate just at the surface of a tumor, not penetrating deep enough. There are two main reasons for that. The first one is dense extracellular matrix that hinders distribution of nanoparticles. The second reason is abnormal blood and lymphatic vessel architecture. It leads to reduced number of blood vessels, to irregular blood and lymphatic flow, and to high fluid pressure. Taken together, all of those aspects effectively block penetration of nanoparticles. To effectively account for those barriers, we need to very carefully design our nanocarriers. Therefore, in EVA Nano project, we are using agent-based model of tumor growth, where we can very precisely represent spatial distribution and composition of all of those barriers in order to simulate how nanoparticles behave in such heterogeneous environment and how effectively they can diffuse in different scenarios.
We're using computer simulations to test how nanoparticle parameters impact their travel through tumour tissue. This includes things such as the size of the particle or how sticky they are, as well as how quickly they're internalised into the cell. We use a combination of agent-based models to work out how the particles travel through the entire tumour, to stochastic reaction diffusion systems to understand how particles move through subsections of the tissue. Right now we're interested in how space matters, that is, how tightly packed in the cells are, and the material around the cells and the impact of that as the particles move through. We're hoping that these computer simulations accelerates the design framework and allows us to work out which parameters matter the most.
One of the main appeals of gold nanoparticles is that we can attach a wide range of different molecules to them and use it to guide the nanoparticles to the preferred target. These attached molecules create a monolayer coating around the nanoparticle, which influences its behavior. Due to the size and physicochemical properties, functionalized nanoparticles can pass through the biological barriers and enter the cells. And the way by which they do this is really important because ultimately this is what determines their function. This process, however, is highly affected by the size, shape, and surface properties of the nanoparticles. So by understanding how it is affected, we can enhance cellular uptake. In EVO-Nano, we perform atomistic molecular dynamics simulations to investigate how the composition of this coating affects the structure in the biological solutions. And then, after obtaining the structure, we can move to a higher level, perform coarse-grained simulations, and investigate how these nanoparticles interact with cell membrane. Results provide guidelines to the more efficient nanoparticle design.
We are dealing with a wide range of scales, from atomistic to whole body scale. Different types of simulations are needed to evaluate at different scales the efficiency of drug delivery by designed nanoparticles. Executing these simulations require a large amount of computational power, but it's not only about how powerful of a computer we have access to. We are looking into finding intelligent solutions to predict the outcome of any given simulations at an earlier stage rather than waiting for the end of the simulations. Our vision is to use explainable AI to understand the intricate inner workings of the interactions of nanoparticles with the environment in which they release the drug they're carrying. We will further use this understanding to evaluate the efficiency of any given nanoparticle design.
We then use artificial evolution to optimize the nanoparticle designs automatically, so we can find new nanomedicine strategies with a click of a button.
By using a simple microfluidic model, we can apply gels that mimic human tissue and then also add nanoparticles that will mimic the nanocarriers of cancer drugs. We can then track these nanocarriers through fluorescent microscopy to gain an understanding of how they might penetrate tissue in the human body.
We are developing artificial tumor microvessel on a chip devices. These devices comprise of microchannel with a diameter in the range of the tumor capillaries and semicircular geometry. Also, they serve as scaffold of human endothelial cells to form an artificial vessel-like endothelium. The devices are used to investigate the transport characteristic of nanoparticle-based anti-cancer drugs in the bloodstream, mimicking as close as possible the human physiological conditions. The microvessel devices are also employed to study how nanoparticles of different sizes or chemistry interact with the vascular endothelium, and how these get extravasated out of the vascular flow to the surrounding tissue into the solid tumors.
The behavior of nanoparticles predicted in silico in EVO-Nano project are then contrasted experimentally. For this, we are using cancer stem cell model in which the cancer stem cells can be visualized and tracked. We are using 2D conventional cell culture as well as 3D models of the tumor spheres and in vivo models. Importantly, we can visualize and count cancer stem cells before, during and after the treatment. We could as well track its behavior since we know that tumor as such tend to compensate for the eliminated cancer stem cells in order to survive.
Our ultimate aim is to use our research to help patients. In order to do that, we need to translate our research results into a marketable product. Therefore, we are starting a new project that is financed by European Commission in the program called FET Innovation Launchpad, where we will first position intellectual property that stem from EVO-Nano in order to develop viable commercialization strategy and reach the market. We will do it through, of course, market research, through research of intellectual property in our domain, and through exploring potential service models, business models, and licensing mechanisms for various stakeholders in both academia and private sector.